Hi, everybody. My name is Jim Shaw, and I'll be talking about our work today, scanning, fast and robust metagenomic sequence comparison. And this is work done with my PhD advisor, Ian William Yu, out of the University of Toronto. This talk will be about measuring sequence similarity for microbial genomes. And the way we tend to do this is we use a statistic called average nucleotide identity, also called ANI or ANI. And this is measured by taking the percentage of nucleotides shared for orthologous regions between two genomes. And in practice, when we use orthologous regions, we tend to be a little less strict. We just really mean a one-to-one alignment between two regions, two genomes. So common use cases of ANI are things like species delineation. Nowadays, we have a 95% ANI threshold for species delineation that has become pretty standard. And we also use it for things like strain level delineation and general measurement of sequence similarity. This talk will be about computing average nucleotide identity rather than its use cases. And to calculate it, I'll go over some of the historic details. So the first methods that calculated average nucleotide identity were using alignment. And what these methods do, they compute alignments using various alignment tools, and then they estimate the sequence identity on regions that they deemed orthologous. So ANI-M, for example, uses the Mummer algorithm as the underlying alignment algorithm, but you can also use things like BLAST and USearch as well as the underlying algorithm. Recently, sketching methods have blown up in popularity, and what sketching methods do is that they obtain a subset of k-mers in the two genomes. Not all the k-mers, but only a subset, and then they use these k-mers to estimate the ANI. And these methods are orders of magnitudes faster than alignments, and they've become very popular because there's been a lot of genomes nowadays, and you have to compute a lot of ANI calculations. MASH and SARMASH are two examples of sketching methods that have become very popular. They have different underlying ways of subsampling the k-mers called either min-hash or frac min-hash, but the idea behind the two are quite similar. And I'll be talking about hybrid methods today, which combine the best of kind of alignment and sketching. So fast ANI is a very popular ANI method, and it uses something called a minimizer min-hash method. So it uses these minimizer k-mers to estimate ANI, but it also uses some sort of mapping as well in order to calculate orthology. The method I'll be introducing today is called skani. We use something called sparse k-mer chaining that I'll dive into later on. But before I dive into our method, I want to talk about metagenomic data. So metagenome assembled genomes, or MEGs, are things we care about computing ANI for, and they're very noisy. And what I mean by this is that they can be incomplete or missing sequences. They don't have all the genomic content in the genome. They can be contaminated, which means that spurious sequences are included that shouldn't be included in the genome. And they can also be fragmented. So you can have small contigs, low N50, and just have a lot of small contigs. And as you can see in this graphic here, as we compute, as we compare incomplete MAGs, you'll have regions that don't overlap fully. And this is just due to missing sequence content. It turns out that mag noise can bias ANI calculations by a lot. So over here, what we have are two identical E. coli genomes with the same nucleotide content, but we've simulated fragmentation and incompleteness by chunking the genomes and removing contigs. And although the ANI should be 100%, you can see that especially fast ANI and MASH, these quicker new methods, they can underestimate the ANI by a lot. Specifically, MASH, as you get more incomplete, as your completeness goes down, the ANI start to go down drastically. Fast ANI, the bias is more due to contig length. So as you get very, very fragmented contigs, fast ANI starts to underestimate ANI quite a lot. So in response to this, we developed skani, a new tool. So skani is a new ANI and align fraction tool. And align fraction here means computing the amount of alignments between the two genomes, what fraction of the genome is aligned. And it works for genomes that are about greater than 82% ANI. It works in MAGs, genomes, even eukaryotic MAGs, and also contigs. We support database search with skani. We use a very fast k-mer ANI filtering method and special indexing data structures that I will talk a little bit about later on. And it's easy to install. So we offer Bioconda installation, static binaries, and it's written in Rust with minimal third-party dependencies. The key points of skani is that, firstly, we use a frac min-hash sketched k-mer filtering algorithm. So really, this is very similar to something like SARMASH. But instead of SARMASH, where you actually compute a final result with it, we only use it as a filter to filter out genomes that are too distant. Once we have filtered out genomes that are too distant, we apply sparse Kamer C-chain mapping. So C-chain mapping is a paradigm used in Minimap2, and it's a way of computing mappings between two genomes. Unlike Minimap2, we've augmented this algorithm because we don't actually need to compute full-on alignments. We just need to compute an ANI. And then finally, we estimate ANI by using unbiased Kamer statistics only on the orthologous regions. And this, importantly, this builds on theoretical work previously explaining ANI estimation bias and seeding. So it turns out if you use certain types of Kamer seeding schemes, you can actually get ANI estimation bias provably mathematically. And these papers have, we took inspiration from these papers to implement a scheme that had minimal ANI bias. So this is a graphic representation of what I just mentioned. So we are comparing these mags. We compute if we can continue if the putative Kamer ANI is greater than some threshold that we set at 80%. And after we deem that these genomes are similar enough, we seed and obtain Kamer anchors with Kamer anchors being matches of Kamers. And then we do this chaining procedure to get these mappings, and then we fragment the genome into chunks, just like other ANI tools. They do this chunking method where we estimate the ANI on each chunk individually, but we don't estimate ANI if a fragment does not have any orthologous chains. In other words, if a fragment has no mapping, then we don't estimate the ANI. This helps us deal with the incompleteness issue. And then we have a distribution of ANI estimates from all the fragments, and we take the mean estimates as our final ANI, and we also output this aligned fraction as well. So skani is more robust than the other methods, and due to our local mapping procedure, and you can see here that it deals with the fragmentation and incompleteness much better on this synthetic data set, but on real data sets, it seems to deal better with it as well. So what we did was we took real mags from this study from Pasoli et al., and we took a bunch of mags with 99%, greater than 99% ANI-M estimate. ANI-M is a robust, slower method, but we took all these mags that seem to be having true 99% ANI, and then we calculated the estimated ANIs for each method. And as you can see here, fast ANI and MASH, they still have this bias issue. As the N50 in the genomes goes down, fast ANI, it starts to go black here, which means that the ANI is being underestimated. MASH, as it gets more incomplete, you can see the darker regions forming, and this is due to, again, underestimation of ANI. Whereas you look at skani, it doesn't show such a pattern. And furthermore, these mag noise issues seem to affect downstream clustering a lot. So you can see here that skani, we're clustering 195 of these mags from the same study, and skani gives very similar results to ANI-M, very similar qualitative results, but you can see these dark streaks forming for fast ANI, MASH, and even SAR-MASH, and that seems to indicate bias due to mag characteristics and not true ANI estimates. Furthermore, skani is very fast, so this is a log scale plot of the speed, and ANI-M takes about 500 times longer than skani. Fast ANI takes about 50 times longer than skani. SAR-MASH and MASH tend to be quite a bit faster. SAR-MASH is only one thread here due to technical issues, but these sketching methods are faster than skani, but skani is still very, very fast. Skani can also do database search. So it turns out that searching an E. coli genome against 85,000 genomes, it only takes about 7 seconds, 6 gigabytes of RAM, and 1 thread. So this can be done on a personal laptop if you wanted to. And searching an entire long-read assembly, which has about 300 megabases of contigs against the same database, only takes less than 2 minutes, 20 gigabytes of RAM, and 10 threads. And this is all due to skani's ability to quickly filter out database matches that are too distant. Something like fast ANI has no such Kamer filter or ANI filter built in, so it would take a lot longer. So the key takeaways are that mags are noisy, and noise induces biases in ANI calculation that could lead to bad downstream results. So MASH incompleteness lowers the ANI a lot, fast ANI, low N50 lowers the ANI a lot, and the alignment methods are good, but they tend to be slow. Skani offers a robust against noise method that leads to better downstream results. A fast method, it's 500 times faster than the mummer-based method, for example, and it can even do database search as well. You can search 85,000 genomes in seconds. So in conclusion, our paper is, this is the title of our paper. We thank the referees for lots of benchmarking advice. It is accepted for publication in Nature Methods as a brief communication now. I'll be presenting the poster at ISMB. And then if you want to check out the GitHub or our paper, these are QR codes linking to it. We'd like to thank the University of Toronto and the National Sciences and Engineering Research Council of Canada for their funding and support. Thank you very much.